Translation is a process where the information on the mRNA sequence is then translated into protein. So mRNA is a sequence of nucleotides. Yep, group of 3 nucleotides will code for a codon. So for example, AUG is one codon, GGC is one codon. So the sequence of these codons on mRNA will determine the sequence of amino acid. So each codon codes for a code for particular amino acid. So this figure shows a dictionary of the genetic code. So the codons of mRNA that specify a given amino acid. So the master code for translation is provided by mRNA codons, so the triplets. So because there are about 64 different triplet codes and there are only 20 different amino acids, so it is not surprising that some of the amino acids are represented by several codons, so which means more than one triplets will code for that specific amino acid. For example, leucine and serine, LEU and SER, yeah, can each be represented by any of 6 different triplets. And only tryptophan, so TRP, so UGG, and methionine, MET, AUG, are presented by a single codon. So in such codons, as leucine, only the first two nucleotides are required to encode the correct amino acid. CU, yeah, for example, leucine here. And the third nucleotide does not change its sense. So this property is called the wobble. This means the wobble will permit some variation or mutation without changing the message, or without changing the amino acids. So one amino acid can be signaled by several alternative codons. So this is called degeneracy or redundancy code. So this allows certain amount of mutation without affecting the ultimate protein produced, as I mentioned earlier. There are 2 types of codons, so the sense codons and the nonsense codon. So the sense codon will code for amino acids. So notice all, but there are 3 nonsense nonsense codons. So the nonsense codons encode for stop codon. So they don't code for any amino acid, but they have a specific message, which is, in other words, stop here. So asking the protein synthesis to stop. So it signals the end of the protein molecule synthesis. The codon AUG codes for the amino acid methionine. Also, this function as the start codon. So the start codon initiates the synthesis of AUG, code for methionine. So it also initiates the protein synthesis. So does all proteins begin with methionine? The answer is no. The initiating methionine is often removed later during the protein synthesis. So where does this process of translation occur in the eukaryotic and prokaryotic cells? So both actually occur in the cytoplasm. So before this, you know that the transcription in the eukaryotic cells occur in the nucleus. The the mRNA, after the transcription, the mRNA is then transported into the cytoplasm, then the translation occur there. But in prokaryotic cells, both transcription and translation happen in the cytoplasm. So this transcription and translation can occur simultaneously. So translation can happen just before the transcription process ends. Now let's have a look at the process of translation. So how protein is synthesized in the cells, all right, through the translation process. But before we start, so it is important to know what are the components of the translation. So to do translation, you need these three components. You need the ribosomal subunit, the ribosome, and then we need the tRNA, which carries the anticodon, right? And also we need the mRNA that carries the message in the form of sequence. The process of translation occurs in 5 stages. The first stage is the initiation, and then we have the elongation, then third we have the termination, fourth is the protein folding, and finally the processing. But for this topic, we only cover or highlight the three important translation process, which are the initiation, elongation, and termination. So in the first step, as you can see here, the picture here, so we have the ribosomal subunit, right? The large subunit that contain the E, P, and A sites, right? And then you see the clove leaf um, structure, that is the tRNA carrying the anticodon. The anticodon, if you can see that there is this UAC there, UAC, and you also have the second anticodon, the second tRNA that contain GAC. So those are tRNA. And then we have on the small subunit, ribosomal subunit, you have the mRNA strand. So that carries the mRNA sequence, right? You have the one starts with AUG and then CUG, blah, blah, blah. And then you have the stop codon, which is the UAG. So in order to start, the first, the correct tRNA, so the correct tRNA that carries the anticodon for the start codon, AUG, will first enter the P site. The P site, and then it will bind with the start codon AUG. So AUG presented in the mRNA. So, so it is important to know that the rules of pairing dictate that the anticodon of this tRNA will complement to the mRNA codon. So AUG. So thus the tRNA with anticodon UAC will first occupy the P site. And then the next step is whereby the second tRNA, you have the second tRNA reading the second codon, right, CUG. So then the second tRNA will fill site A, all right, and then you have now two tRNAs carrying the amino acids, so that code by the mRNA sequence. And then in the the next step is formation of peptide bond. So you have two amino acids, right? So the entry of the tRNA2 into site A brings the two adjacent tRNA in favorable proximity for a dipeptide bond to form between amino acid 1 and amino acid 2. So the first, the methionine is transferred from the first tRNA to amino acid 2, so resulting two couples of amino acid, which call a dipeptide. Okay, so now you have two amino acids, and then the bond is formed, a dipeptide bond is formed. So now you have two amino acid, which is called dipeptide. For the next step to proceed, some room must be made on the ribosome, right? Because you have only two sites of P and A sites where the RNA can can bind, right? So the next codon in the sequence must be brought into the position for reading. So this process is accomplished by a process called translocation. So this is an enzyme directed shifting of the ribosome to the next position of the mRNA strand, so which causes the blank tRNA 1 here at the P site to be discharged from the ribosome at the E site. So the tRNA first from P site move to the E site. Then you have the amino acid is being removed from the tRNA, right? So the next step is where um, first translocation, the fourth step. So first translocation, tRNA2 shift into the P site. tRNA3 will enter ribosome at A site. So now after the P site is empty, right, the second tRNA actually will move from A site to the P site, leaving A site empty. So now the A site is ready for the insertion of tRNA3 at A site as directed by the third mRNA codon. So the peptide is then formed between between dipeptide and the third amino acid. So now we have three amino acids making tripeptide, right? And then the next one is the process where the peptide is split. So splitting of the peptide from tRNA2. So the peptide, the amino acid, the peptide now is split from from tRNA2, and the translocation process will occur. So that is actually the same process as the previous one, how the amino acid is detached from the tRNA. The splitting of the tRNA2 with the dipeptide, so now releases tRNA2 to in the E site, right? So this shift mRNA to the next position. So the tRNA3 then will move to P position. So initially it was from A and then it moves to P position. So now opens up, when it moves, then opens up A site is empty now, so that tRNA4 can be brought in. So from this point, peptide elongation proceeds repetitively by the same reaction of actions out to the end of the mRNA until uh, until the ribosome reach at the stop codon UAG here. Then the protein synthesis will be terminated. So that is overall the process of the translation. In the process of metabolism, the metabolic reactions in cells um, involve enzymes, right? So enzymes are actually regulated, and that one form of regulation occurs at the genetic level. So this control of mechanism ensures that the genes are active only when their products are required. So in this way, enzymes will be produced as they are needed, and the waste of energy and materials in dead end synthesis will be prevented. So a major form of gene regulation in prokaryotes is through system called operon. So you're going to learn about operon. So for the metabolic reactions in the pathway, so it needs enzyme. Bacterial cells only produce those enzymes for the pathway when they are substrates. So this is called enzyme induction. So the bacterial cells will selectively manufacture the enzymes that are required for the uptake and the metabolism of the nutrients when substrates that are present in the medium. So the substrate will induce the synthesis of enzyme. For example, the metabolic pathways for the galactose catabolism, lactose and arabinose catabolism in E. coli. So in normal condition, for example, you have a nutrient in the presence of glucose. All these enzymes for catabolism of lactose, galactose will not be produced. So when lactose is present in the medium, then only those enzyme is produced. An operon is a section of DNA that contains one or more structural gene along with the corresponding operator gene that control the transcriptions. So having this kind of setup will permit genes for a particular metabolic pathway to be induced or repressed in unison by the same regulatory element. The clusters of genes are controlled at one promoter site. So this is examples of operons. We have the GAL genes, operon that code for the LAC genes, and the ARA genes. These are all for different enzymes. And we have the promoter sites that control the expression of these enzymes. Operants are described as either inducible or repressible. So the category of an operon is determined by how it is affected by the environment within the cell. Many catabolic operons are inducible operon, so meaning that the operon is turned on or induced by the substrate of the enzyme for which the structural gene code. So in this way, the enzymes needed to metabolize a nutrient, for example lactose, are only produced when that lactose or when that nutrient is present. The second type of operon is the repressible operons. So these operons often contain genes encoding for anabolic enzymes, such as those used to synthesize amino acids. So in the case of these operons, several genes in series are turned off or repressed by the product synthesized by the enzyme. In the operon, there are processes called induction and repression. So induction is when the transcription of the gene turns on. So inducer is the substance that act to induce the transcription of the gene. And the default position of the inducible gene is always off. So the second condition is the repression condition. So during this repression, so it inhibits the gene expression. So the protein synthesis will be decreased. So this process is a response to the overabundance of an end product. So when there is a lot of the end product, then the repression will occur. So this process is mediated by a regulatory protein called the repressors. So the repressors will block the ability of the RNA polymerase to initiate the transcription from the repressed gene. So the default position of a repressible gene is always on. The lack of nutrient can also induces the synthesis of enzyme. For example, the enzyme for the production of the amino acid tryptophan. In the absence of tryptophan, then the enzymes are synthesized. So the enzyme to produce the tryptophan is synthesized because there's no tryptophan in the environment. So the cells need tryptophan. When the tryptophan is produced, when it is synthesized, then the synthesis of the enzyme is then repressed because you have now a lot of tryptophan in the cells. Now let's have a look at the operon model for the gene expression. We're looking at the inducible operon. So the best understood cell system for explaining control through the genetic induction is the lactose or lac operon. So the system was first established or described by Jacob and Monod in 1961. So uh, this system accounts for the regulation of lactose metabolism in E. coli. So many other operons with similar modes of action have since been identified, and together they furnish convincing evidence that a cell's metabolic actions can have great impact on gene expression. So let's have a look at the lactose metabolism in E. coli. So it is an inducible system. So the main enzyme in this system is the beta-galactosidase. Beta-galactosidase that splits lactose into glucose and galactose. So when E. coli is placed into medium with no lactose, for example, just glucose, so this beta-galactosidase will not be produced. But when lactose is present, the beta-galactosidase is produced. So the lactose acts as an inducer to the system or to the lac operon. The lactose operon has three important features. The first one is the regulator that is composed of the gene that codes for a protein capable of repressing the operon. So the gene is called a repressor. The second one is the control locus. So this control locus composed of two areas. Number one, the promoter that recognized by the RNA polymerase, and then the other one is the operator. Operator is a sequence that acts as an on and off switch for the transcription. The last feature is called the structural locus. So this structural locus in the lac operon is made up of three genes, so each encoding for different enzyme needed to catabolize lactose. So one of the enzyme is beta-galactosidase, which hydrolyzes the lactose into glucose and galactose into the monosaccharide, and another one is permease that bring lactose across the cell membrane, so bringing in the lactose from the medium into the cells. And the other one is the transacetylase. Transacetylase is the enzyme that metabolizes the disaccharide other than lactose. So this is basically all the structural genes in the lac operon. So we have the promoter, that regions of DNA where RNA polymerase will start the transcription. The operator will go or stop signal for transcription of the structured gene. And then we have the repressor that repress the gene when lactose is not present. So let's have a look at the two different conditions, when the lactose operon is off and when the lactose operon is on. So in the absence of lactose, as when there is no lactose in the medium, so that means the operon is turned off. Okay, so what happened during this process? The repressor protein will bind to the operator site. So the operator site, so then this will prevent the RNA polymerase from transcribing the adjacent structural genes. So therefore, there will be no mRNA is made, and there will be no enzyme synthesized. Okay, so save the energy of the cells, so you don't have to produce these beta-galactosidase enzymes. This is the condition for the lac operon is turned on. So when lactose is present, so you have lactose in the medium, then the operon turn on. So where in this process, the repressor protein is inactivated by the lactose. So in opposite for the previous condition where it is turned off, no lactose, so the repressor protein is active, right? So it is active, then it binds to the operator region. But in this condition, uh, lactose is first transported into the cells and then converts into an inducer called allolactose. So the molecule from lactose is converted to an allolactose. So now it is ready to be used as the inducer. So this allolactose actually binds to the repressor protein. So when it binds to the repressor protein, this repressor cannot bind to the operator site, leaving the site free. Right now, the RNA polymerase uh, can bind to the promoter and then run through the DNA so that it allows the transcription of the DNA into mRNA, and then those mRNA will produce protein that encode for the enzyme for the lactose processing. Okay, the second model of gene expression is the repressible operon. So the operon that is used as a model is the tryptophan operon. So the genes for enzyme synthesis of tryptophan, the amino acids. The structural genes are transcribed and translated, leading to tryptophan synthesis. So in this process, it is a process of amino acid anabolism to produce the tryptophan. However, when excess of tryptophan is present, when there's a lot of tryptophan in the cells, the tryptophan will act as a co-repressor. So this co-repressor will bind to the repressor protein. So when the repressor protein is bind, then it will bind to the operator, stopping from the tryptophan synthesis. When there is no tryptophan in the medium, so you have a repressor protein that is in inactive form. So when they are inactive, it cannot bind to the operator. It won't bind to the operator. So then the RNA polymerase can initiate transcription, and then you have now all the protein for the tryptophan synthesis. On the opposite condition, when tryptophan, you have a lot of tryptophan, the tryptophan will bind to the repressor protein and make it active. So now the repressor is now active. It now binds to the operator region. So when the tryptophan is activating it so that it can bind to the operator. So now the structural genes cannot be transcribed. So this means that the operon is turned off when there's a lot of tryptophan in the medium. So that's all for the operon topic.